Welcome to another episode of BioBizBuzz, the podcast where we explore the cutting edge of innovations that are shaping the future of biopharmaceuticals. I'm your host, Mike Ward, and I'm also the Global Head of Life Sciences and Healthcare Thought Leadership at Clarivate, which is a multinational provider of insights, intelligence, and data that are helping the industry accelerate its innovation. In this episode, we're going to be diving into one of the most exciting frontiers in drug discovery, that is targeted protein degradation. And to help me with this, I've been joined by Per Scholtblad, who is the co-founder and CEO of Beactica Therapeutics, which is a Swedish biotech that's focusing on targeted protein degradation. So let's consider the background. The pharmaceutical industry faces formidable challenges as traditional drug development approaches actually struggle with what are called undruggable targets. These are proteins that lack well-defined binding pockets for sort of the conventional small molecule inhibitors. Compounding this, resistance mechanisms continue to emerge, rendering actually many of the existing therapies actually ineffective over time. Perhaps nowhere are these challenges more acute than actually in cancer therapeutics, particularly for devastating diseases like glioblastoma, where the median survival rate is just 12 to 8 months, despite decades of research effort. However, emerging technologies are beginning to offer new hope. Targeted protein degradation represents a revolutionary shift from the traditional inhibition approaches by actually harnessing the cell's own quality control machinery to eliminate disease-causing proteins entirely. This event-driven strategy that can potentially overcome many limitations on the conventional drugs, including the ability to target previously undruggable proteins, as well as counteracting resistance mechanisms. Indeed, at Clarivate, we recently published a Companies to Watch report focused on biotechs who are active in the space, but may still be not on the radar of most pharma companies or investors. One of the biotechs at that cutting edge of science that was featured in the report is in fact free to download, is Beactica Therapeutics, which has positioned itself at the forefront of this revolution with its proprietary Eclipsor platform. It's a platform that enables the development of both allosteric modulators and targeted protein degraders. But more importantly, the company's lead program, BA17, represents a first-in-class approach to treating glioblastoma by targeting the LSD1 CoREST complex, a strategy that could meaningfully extend survival of patients with this uniformly fatal disease. So, Per, thanks for joining us on BioBiz today. So let's start with the big picture. The pharmaceutical industry has long struggled with these so-called undruggable targets. Can you explain what makes a protein undruggable and how Beactica's approach is addressing this fundamental challenge?
Yes, thank you first, Mike, for inviting us onto this podcast. The term undruggable has been used in the pharmaceutical industry for decades. It's been estimated that up to 85% of the human proteome is undruggable. Many of these targets lack well-defined binding pockets, have very shallow binding sites, and often have complex roles beyond simple enzymatic activity. For example, they may mediate their function through protein-protein interactions or act as structural scaffolding components in multimeric protein complexes. Traditional drug discovery focused on inhibiting active sites of enzymes or receptors, essentially equivalent to jamming a lock. But what if the protein doesn't have a proper lock to jam? This is where our Eclipsor platform comes in. We take two complementary approaches. First, our allosteric modulators bind to sites that are distant from the active sites, inducing conformational changes that modulate protein function. Secondly, our targeted protein degraders don't just inhibit, they eliminate the entire protein by tapping into the cell's natural waste disposal system. One of the beauties of allosteric modulation is that these sites often are much less conserved across protein families. which provides additional opportunities to achieve a more exquisite selectivity. And unlike traditional inhibitors that compete with natural substrates, allosteric modulators can work also when the natural ligand is present. This offers a more nuanced possibility to control protein function and modulate biological pathways.
Yeah, you mentioned that the Eclipsor platform is focusing on both allosteric modulators and targeting protein degraders. For our listeners who may not be familiar with these approaches, can you break down how they differ from, say, traditional drug discovery methods and why they're actually particularly suited to addressing these challenging targets?
Yes, traditional drug discovery is like trying to stop a machine by blocking its main gear. Our Eclipsor platform offers two fundamentally different approaches. With allosteric modulators, we're essentially finding the machine's hidden control panel to finally adjust protein activity, allowing for more nuanced control than simply switching a protein on or off. Allosteric modulation is achieved by targeting binding sites that are structurally and functionally different from where the natural substrate binds. This gives us several advantages. We don't have to compete with a high concentration of natural substrates, and we can typically also achieve a different level of selectivity because allosteric sites are often unique across protein families. Allosteric modulation also offers better opportunities to fine-tune the target protein activity rather than just switching it on or off. It can be thought of as a lighting dimmer as opposed to an inhibitor, which is a simple old-fashioned on-off switch. In contrast, our protein degraders take a more radical approach. Instead of just inhibiting the machine, we remove it altogether from the factory floor. This is particularly powerful for multifunctional proteins that serve scaffolding or regulatory roles beyond a simple enzymatic activity. When you degrade a protein, you eliminate all of its functions in one blow. Degraders also have the advantage of their catalytic effect, where a single degrader can tag multiple copies of the target for destruction. Compared to a traditional inhibitor that only blocks a single target, the degrader's so-called event-driven pharmacology provides superior potency and can be efficacious at much lower doses. What is making Eclipsor unique is our ability to use the synergies across these two modalities and precision engineer solutions that matches the need in a given biological situation. For example, an allosteric binder can be an excellent target ligand forming part of a bifunctional degrader. With Eclipsor, we don't just discover compounds, we systematically design them based on deep understanding on protein function, structure, and dynamics. This integrated approach allows us to tackle challenging targets where others see significant limitations.
Yeah, so I mean, that's fascinating. As I mentioned in my introduction, glioblastoma remains one of the most devastating cancers with survival rates that haven't been meaningfully improved in the decades. You've got a lead compound, BA17, which targets the LSD1 co-rest complex in this indication. What makes this target particularly promising for glioblastoma, and how does protein degradation offer advantages over traditional inhibition approaches?
Glioblastoma is devastating precisely because these tumors have mastered immune evasion. This is a mechanism that cancer uses to hide from the body's own defenses, making tumors very hard to treat. The LSD1 co-rest complex plays a central role in this process by silencing genes involved in antigen presentation and immune recognition. LSD1 achieves this silencing by controlling the methylation status of proteins. This includes histones, but also other proteins involved in transcriptional regulation. BA17 doesn't just inhibit the complex, it degrades both LSD1 and its binding partner co-rest. And this is crucial because LSD1 has multiple functions beyond its demethylase activity, including stabilizing other transcriptional regulators. By degrading the entire LSD1 co-rest complex, we simultaneously restore antigen presentation, induce viral mimicry responses, and we also reprogram tumor-associated macrophages towards a pro-inflammatory state. The degradation approach offers a key advantage over inhibition. Even if some LSD1 molecules escape inhibition, degradation provides a functionally broader effect that is also more durable. And this is particularly important in the harsh tumor microenvironment where drug penetration and target engagement can be very challenging.
Yeah, sure. And challenges are clearly something that need to be addressed. In our recent report, we sort of noted that the targeted protein degradation field has seen explosive growth, with the market projected to reach $5.5 billion by 2034. However, the field still faces some significant technical challenges, whether it's the hook effect at high concentrations or issues around oral bioavailability. How is a company like Beactica addressing these industry-wide challenges in your drug development programs?
Yes, Mike, you're absolutely right. The targeted protein degradation field faces several technical hurdles that we've systematically addressed in our programs. Oral bioavailability is probably the biggest challenge. Traditional protacs tend to be large, complex molecules that violate Lipinski's rule of 5. We're addressing this through our platform's ability to design more drug-like molecules from the ground up, rather than simply linking existing inhibitors to E3 ligase binders. The hook effect is a well-known challenge that happens at high protac concentrations. Instead of promoting degradation, the molecule saturates their individual binding sites on either the target protein or on the E3 ligase, preventing the formation of the essential ternary complex required for the effective degradation of the target. As a result, degradation efficiency decreases at these very high concentrations. We're engineering our degraders to have optimal ternary complex cooperativity and avidity, which extends the concentration window where degradation remains efficient. Our medicinal chemistry team uses sophisticated data-driven design to optimize the linker region and binding geometries to favor formation of productive ternary complexes.
Right. In the sort of the Clarivate report that we published, I was interested to sort of see in your profile that you've established some pretty impressive academic collaborations with institutes like the Uppsala University, the Karolinska Institute, and MD Anderson Cancer Center in the US. In a field moving as rapidly as targeted protein degradation, how critical are such partnerships to actually staying at the cutting edge of the science?
That's a good question, Mike. In a field that is moving as rapidly as targeted protein degradation, no single organization can master all the required expertise. Our collaborations with Uppsala University, Karolinska Institute, MD Anderson, and the Broad Institute, etc., gives us access to cutting-edge insights that are often 12 to 18 months ahead of publication. These partnerships are particularly valuable for understanding target biology in disease contexts. For example, our collaboration with the university in Leuven specifically focuses on glioblastoma applications of our LSD1 co-rest degrader, whilst our work with MD Anderson explores T degraders in different cancer contexts. Each institution brings unique patient samples, disease models, clinical insights that inform our drug design. The academic partnerships also help us to stay ahead of emerging resistance mechanisms and combination opportunities. In the target protein degradation field, understanding how cells adapt to protein loss is crucial for designing durable therapies. Our academic collaborators often identify these mechanisms before they become clinically apparent. And equally important, these collaborations help validate our platform's versatility across different target classes and disease contexts. So when multiple independent groups can successfully apply compounds developed with our Eclipsor platform to their research problems, it demonstrates the robustness of our approach.
Yeah. So looking at your TEAD program, P65047, you've mentioned superior efficacy compared to competitive compounds and a remarkable correlation with the VGLL3 regulated transcription. TEAD proteins have emerged as validated targets for hippo-altered cancers. So can you elaborate on what actually makes your approach to TEAD degradation unique?
TEAD transcription factors are validated targets in Hippo pathway-altered cancers, but most approaches focus on disrupting the YAP-TEAD interaction, either by inhibiting the auto-palmitoylation of TEAD required for activation, or by directly blocking the YAP binding interface of the TEAD protein. Our compound P6547 takes a fundamentally different approach by degrading TEAD entirely. What makes this exciting is the remarkable correlation we observe between P6547 efficacy and VGLL3 regulated transcription. VGLL3 overexpression is associated with poor patient outcomes in multiple cancer types, and we've discovered that VGLL3 can actually confer resistance to traditional TEAD inhibitors by activating alternative survival pathways, including SOX4 PI3K ACT signaling. And by degrading TEAD proteins entirely, we eliminate not just their transcriptional activity, but also their scaffolding functions and ability of resistance-mediating cofactors like VGLO3 to exert any effects. Our degrader shows superior efficacy compared to compounds across multiple cancer lines, suggesting we're hitting pathways that other approaches miss. The dose-dependent elimination of TEAD1 that we observe in target tissues, combined with a VGLL3 correlation, gives us a potential biomarker strategy for patient selection. We can identify patients whose tumors are likely to be more sensitive to TEAD degradation versus those who might benefit from combination approaches, for example.
Right. Again, really interesting. So I'd like to sort of revert back to your lead program, BA17. The LSD1 target has attracted significant attention across the whole of the industry, with many companies developing both inhibitors and degraders. So what is actually differentiating Beactica's LSD1 Corex degrader approach from those other programs? And why do you believe that degradation actually offers advantages over inhibition in this target?
Yes, the LSD1 field is crowded with many companies developing catalytic inhibitors, but to the best of our knowledge, none are focusing on degraders. One of the challenges is that LSD1 has many roles that changes with its specific binding partner in each context. For example, when bound to co-rest, it demethylates one specific histone lysine, while in complex with the androgen receptor, it demethylases another lysine on the same protein. And this sort of fine-tuning leads to nuanced differences in the downstream biology. The catalytic inhibitors, which have been dominating the field, block the demethylation step, but leave LSD1 in place to perform the structural roles within these complexes, which often means that you need combination therapies to see an effect. Our degrader, BA17, takes a different approach by removing LSD1 itself. This breaks down both the co-rest associated and other LSD1 dependent complexes, leading to a functionally selective shutdown of the pathway. Whilst this approach isn't complex selective, it avoids some of the off-target issues seen with older inhibitor, and it removes the need for example, HDAC inhibitor co-therapy. Furthermore, BA17 has been optimized for favorable pharmacokinetic properties, achieving both oral bioavailability and high brain exposure, key challenges that have limited the success of many previous LSD1 targeted approaches. The FDA orphan drug designation for BA17 in glioblastoma underscores the urgent medical need in this disease and the therapeutic potential of our approach in this very devastated condition.
So thinking about that sort of FDA orphan drug designation that you've got for BA17 in glioblastoma treatment, as you advance towards phase 1 readiness, what are the key biomarker strategies that you're implementing to enable precision patient selection and actually maximize the probability of clinical success?
Well, our biomarker strategy focuses on identifying specific gene activity patterns and circulating markers in the blood to see if our drug is working and to identify patients most likely to benefit. We begin by analyzing large datasets of genome expression profiles, both from advanced animal models and patient tumor biopsies. This allows us to develop companion diagnostic tests that can predict which patients are most likely to benefit from treatment with BA17. We're also implementing circulating biomarkers to monitor target engagement and pathway modulation. As BA17 degrades the LSD1 CoREST complex and reactivates immune genes, we expect to see changes in circulating immune markers and cytokine profiles that correlate with antitumor activity. Perhaps most importantly, we're developing functional biomarkers that predict which patients are most likely to mount effective immune responses once the epigenetic breaks are released. Not all glioblastoma patients will have immune systems capable of responding, even with an optimal target degradation. So identifying those patients up front will be crucial for clinical success.
Yeah. And the target protein degradation field is clearly rapidly evolving. So, we're seeing new modalities beyond PROTACs emerging, which include the molecular glues or novel E3 ligase recruitment strategies. So how do you see the competitive landscape evolving, and what advantages, for example, does your platform-based approach actually provide in this dynamic environment?
Yes, the targeted protein degradation field is progressing very quickly indeed, moving past traditional PROTACs, embracing strategies such as molecular glues, lysosome-directed chimeras, and innovative methods for E3 ligase recruitment. Our platform approach positions us really well for this evolution. So rather than being locked in a single degradation modality, our Eclipsor platform can incorporate new degradation mechanisms as they emerge. We're already exploring alternative protein disposal pathways beyond the proteasome. The key competitive advantage is our integrated understanding of target biology, allosteric modulation, and degrader kinetics. Many competitors excel in one of these three arenas, but struggle to integrate all three. Our track record, spanning more than 15 years, demonstrates that our mechanistic innovation and understanding of complex protein science successfully can be translated into clinical candidates. And looking ahead, the ability to adapt quickly to emerging degradation modalities will become a defining advantage for companies in this field. Success will increasingly favor those who can deliver both multiple clinical stage programs and also integrate novel degradation approaches into their work. Our diversified pipeline, spanning various target classes and therapeutic areas, and our flexible platform enable us to incorporate these advantages seamlessly, gives us a clear position at the forefront of this very rapidly evolving landscape.
Okay, yeah. So you mentioned there diversified pipeline. So, looking toward the future and without, I guess, revealing any proprietary information that you don't want to share, can you sort of explain to us what your strategic thinking is around both target selection and also therapeutic areas that you might be expanding into?
Our target selection strategy focuses on three key criteria: high unmet medical need, platform advantage, and finally, well-defined patient populations. We prioritize targets where allosteric modulation and degradation provides genuine advantages over existing approaches, not just alternative routes to the same endpoint. This typically means multifunctional proteins, scaffolding proteins, proteins involved in multimeric complexes, or targets involved in resistance mechanisms. Our therapeutic area expansion follows the science rather than market considerations. If our platform can address a compelling biological target in a rare disease when no treatment options are available, we'll pursue it even if the commercial opportunity is modest to start off with. Conversely, we won't chase large market if we can't deliver genuine patient benefit. The undisclosed programs in our pipeline reflect this philosophy. We're targeting proteins that others consider undruggable, but where our platform's unique capabilities create real opportunities for breakthrough therapies.
Yeah. And for biotech investors and industry observers, what are the sort of the key milestones that we should all be watching out for from Beactica over the next 18, 24 months? And how do you see the company's trajectory in the broader targeted protein degradation landscape?
Over the next 18 to 24 months, investors should watch for several key inflection points. First, IND filing for BA17 in glioblastoma, which will validate our degrader platform clinically. Secondly, indication selection and completion of lead optimization for our T degrader P6547. We're also advancing our CNS collaboration with BioArctic, which could open an entirely new therapeutic area for our platform. The Eclipsor technology's applicability to neurodegeneration represents a great expansion opportunity for us. From a business development perspective, we expect to see increasing interest from large pharmaceutical companies seeking to access our platform capabilities. The targeted protein degradation space is consolidating, and companies with proven platforms and multiple clinical stage assets will command premium valuations. Our trajectory makes us an attractive platform acquisition or partnership opportunities for companies wanting to establish TPD capabilities quickly rather than building them from scratch. With $32 million raised to date and several value-driving milestones ahead, we're well positioned for our next stage of growth.
Yeah. Okay. So, as we sort of draw to a conclusion, thinking about patients who are going to be ultimately served by these breakthrough technologies, what gives you the most confidence that your approach will actually deliver meaningful clinical benefits for people facing life-threatening diseases?
What gives me the greatest confidence is our deep integration of target biology with platform technology. We start with unmet medical needs and work backwards to identify targets where degradation offers genuine biological advantages. For glioblastoma patients, the problem isn't just tumor growth, it's immune evasion. Our LSD1 CoREST degrader directly addresses the root cause by reactivating the immune system's ability to recognize and attack cancer cells. Our track record of delivering pharmaceutical innovation over more than 15 years gives me confidence in our capabilities. We have successfully enabled the translation of complex protein science into clinical candidates before, and our platform approach means each new program benefits from accumulated learning. Most importantly, we will maintain our focus on the greatest medical needs. Glioblastoma patients have a median survival measured in months. Even modest improvements in their outcomes represent profound victories for patients and families facing this devastating disease. That patient focus drives everything we do, and it ensures that we're developing therapies that truly matters. Finally, Mike, I would like to thank you so much for inviting us onto BioBizBuzz today. It's been an absolute pleasure speaking to you.
Well, I think that's a very fitting way, a very positive way to actually wrap up this episode of BioBizBuzz. So my thanks to Per for sharing insights into Beactica's innovative approach to both targeted protein degradation and its mission to transform the prospects for patients with life-threatening diseases. The targeted protein degradation field definitely represents one of the most promising paradigm shifts in modern drug discovery. It's offering new hope for tackling previously intractable targets, as well as overcoming resistance mechanisms that have long plagued the traditional therapeutics. Companies like Beactica, with their validated platform technologies and strategic focus on those high unmet needs, those indications that are difficult to treat, are clearly positioning themselves to lead in this therapeutic revolution. So if you found this episode interesting, please leave us a review and subscribe wherever you get your podcasts. And don't forget to turn on your push notices so you don't miss future episodes of BioBizBuzz, where we keep you informed of the innovations that are shaping the future of medicine. My name is Mike Ward. Keep on innovating, and goodbye until the next episode of BioBizBuzz. Thank you.